Welcome to the Deep Dive, the show where we take this just a vast and often totally contradictory landscape of human knowledge, from ancient metaphysics all the way to cutting-edge lab science, and we try to find the hidden connections that, really govern our world.
And today, I think we've given ourselves a pretty monumental task. We are trying to understand the fundamental laws of persistence.
Persistence.
It's
yeah.
not just about survival. The real mystery, I think, is how organized structure, life itself, actively resists the most powerful force in the universe.
Entropy.
Entropy. That universal slide toward total disorder and chaos.
And the sources you've curated for this deep dive are, they're truly stunning in their scope. We're going from the chaotic primordial soup of early Earth. We're talking about these really sophisticated thermodynamic models for how life started.
And then we pivot completely.
Completely. We jump to a 5-minute South Indian yoga routine that's designed for longevity, and then we finish in this just absolutely bizarre world of regenerating flatworms that can literally grow 2 heads on command.
It sounds like half a dozen different topics, I know.
Yeah.
But the core question running through all of them is unified. How does organization persist? And we're looking at 2 really distinct, but I think profoundly related frameworks that try to answer that.
Okay, so the first one is, it's all about physics, thermodynamic and physical persistence.
Right. This framework sees the origin of life abiogenesis, not as some, miracle, but as a necessary, almost inevitable consequence of energy just seeking the most efficient path to flow and dissipate.
It's hard physics basically determining destiny.
Exactly. And then paralleling that, we explore the framework of biological and energetic persistence.
And this one is drawn from, ancient philosophy and modern biophysics.
A combination, yeah. It views longevity and aging as a system that has to consciously and unconsciously manage its own internal energy, what one of our sources calls biomagnetism, to maintain its physical form and, crucially, the integrity of its consciousness.
So the mission for this deep dive, for you listening, is to realize that the universal principles that governed the very first self-organizing chemical reaction on Earth, they might be the exact same principles we have to master today to fight aging and extend our own persistence. It's all about energy.
Energy, alignment, and flow.
Okay, let's unpack this. Let's start at the very beginning, at the dawn of structure, and look at the physical laws that sort of demanded life emerge.
We have to start with the historical problem. The issue that traditional origins of life chemistry has faced for decades. For a long time, the cornerstone of the whole field was the Miller-Urey experiment.
Right, that's the famous one.
It proved the fundamental plausibility of generating the basic building blocks of life, amino acids, from very simple precursors just by simulating early Earth conditions.
They took some water, methane, ammonia, hydrogen, zapped it with electricity.
And voila.
You get the building blocks of proteins. That must have felt like a massive breakthrough. Like life was just a matter of random chance and a lot of time.
It was incredibly inspirational. But that success, it kind of led researchers down a specific path. They started trying to synthesize every other important biomolecule, ATP, lipids, DNA, RNA in the lab using similar conditions.
And that's where they hit a wall.
A massive wall, an almost insurmountable natural feasibility problem.
Which was the problem of specificity, right?
Exactly. They discovered this wide variety of vastly different, often mutually exclusive conditions that you needed to successfully make each different molecule.
So to make one thing, you might need, high temperatures and an alkaline environment.
Right. And then for the very next step in the chain, you suddenly need intense UV light, an acidic pH, and maybe a wet-dry cycle to get the polymers to condense.
And you're talking about a chemical system that needs perfect timing, perfect temperature, perfect pressure, all of it lining up one after the other, totally spontaneously in this giant, messy, ancient ocean.
The probability seems astronomically low.
It's questionable, to say the least.
Yeah, it really is. And that's why people like the biochemist Christian de Duve, among others, suggested a really radical shift in perspective. He said, instead of focusing on these isolated reactions, which assume the environment has to do all the work,
Providing these perfect little steps.
right, he said, we should look at life processes themselves. Things like autocatalytic cycles and evolution. We need to stop looking at just the molecules and start looking at the physics of the process itself.
And that shift is what brings us to this new thermodynamic definition of life's emergence. It's one that doesn't need this, miraculous sequence of chemicals. It suggests life is almost a thermodynamic inevitability.
This is the idea that natural selection isn't just about genes, it's about energy regulation. It's about enabling sustained, persistent reaction cycles.
Which is the core of the thermodynamic abiogenesis likelihood model, or TEL.
Right. And what makes TAN so radical is that it's completely molecule agnostic. It doesn't care if it's RNA or peptides or something we haven't even thought of.
It just sees life initially as a self-sustaining network of reactions.
Yes. All it cares about is that the network persists.
Okay, so if the system doesn't need heredity or replication to get started, what defines selection? How does one reaction network, beat out another one?
Selection is redefined. It's purely a consequence of persistence. The sheer ability of a reaction network to just keep itself going over time, driven only by thermodynamic constraints in a non-equilibrium environment.
So the systems that last the longest are the ones that are selected.
That's it. It's survival of the most stable.
Let's deconstruct the core engine of this model because it's based on a surprisingly practical idea. It's like an energetic accounting system. The reaction viability inequality.
It is. It's the budget that dictates survival. For any system to persist, its net energy budget has to meet or exceed the total energy cost to sustain all its internal reactions.
So it's an equation.
It's a balance. Mathematically, it balances the energy coming in from the environment, which they call ZLA, that's your fluctuating external energy, like heat from a vent or sunlight.
Okay, so environmental energy plus the energy the system itself releases from its own reactions. That's $3.
Right, so the total available energy is zillion dollars plus the sum of all the dollars, and that total has to be greater than or equal to the sum of all the $6.
And $6 is the cost.
Yeah.
The energy needed to drive the reactions.
Exactly. It's the maintenance cost. If your inflow drops below your maintenance cost for too long, the whole system just collapses back into disorder.
That seems pretty basic for any system. But TALEM introduces the real secret sauce for persistence, which is the internal management of energy, the ability to store it.
This is the critical component. They call it $6. Persistence is profoundly governed by how the reaction network can internally release or, crucially, store energy. And that stored energy, whether it's in chemical bonds or some kind of structural asymmetry, it acts as a buffer.
It's the prebiotic battery backup you mentioned earlier.
Precisely. If the environmental energy temporarily dips low, a hydrothermal vent cools down for a bit, or a puddle dries out, a system that can tap into its stored internal energy, its $6, can keep its required reactions going and just survive the fluctuation.
So the systems that survive these ups and downs, the ones that accumulate this structural energy,
Yeah.
they're the ones that get naturally selected for persistence. And this is long before DNA or cell walls even existed.
Exact.
This reframes the whole fight against entropy. We think of entropy as disorder, the enemy of life. But
Right.
here, the sources are suggesting that entropy, paradoxically, can actually be a driver of this localized order. How does that work?
Well, Thallium posits that in open systems that are far from equilibrium, and that just means there's a constant high energy flow through them,
Like the early Earth.
like the early Earth. In those systems, entropy isn't the enemy, it's the engine of diversity. The energy fluxes, those thermal gradients, those wet-dry cycles, they dramatically increase global entropy. The messiness of the universe goes way up. But in doing so, they simultaneously enable and stabilize localized chemical order. They make it possible for these transient, persistent structures to form.
It's the ultimate paradox. The universe wants to be messy, and the most efficient way for it to get as messy as possible is to create these highly organized, tiny little pockets of persistence life to speed up the dissipation of energy.
And this aligns beautifully with some historical thermodynamic concepts. I mean, we're talking about Ilya Prigoghine's idea of dissipative structures.
Where ordered systems can just arise spontaneously because they're better at dissipating energy.
Exactly. Prebiotic systems that achieve this crucial balance between energy coming in and the energy their reactions require are just. They're naturally self-stabilizing.
And Jeremy England's concept of dissipative adaptation fits right in here too, right? The systems that statistically evolved to dissipate energy more efficiently are the winners.
Absolutely. Selection in that prebiotic era wasn't about who had the best genes, because there were no genes. It was about who was the most efficient thermodynamic conduit. The reaction chains that survived the fluctuations and maintained their structure just naturally dominated the landscape.
This brings us to a fascinating hypothesis that connects this, pure thermodynamics back to the actual molecules of life,
Yeah.
RNA and DNA. The sources suggest their first purpose was purely thermodynamic, not informational.
They are powerful thermodynamic structures. The research shows that RNA and DNA are incredibly efficient absorbers of intense ultraviolet light.
Specifically the high-energy UV that was blasting the early Earth because there was no ozone layer.
Right, in the 200, 300 nanometer range. And crucially, they are also remarkably rapid at dissipating this high energy almost instantly into harmless heat, as long as they're in water.
So their primary function was basically a highly efficient molecular sunscreen and an energy sink. They weren't blueprints, they were dissipators.
And this leads to the hypothesis that life began and persists as this massive global catalyst for absorbing and dissipating intense sunlight, especially on the surface of the Archean seas.
Okay, I get the local benefit. It protects the chemistry happening just below the surface. But how does that scale up to the entire planet?
Well, the thought is that by absorbing all this UV radiation and converting it into lower-grade heat, they promote coupled, irreversible processes. They fundamentally speed up the Earth's energy cycle.
You mean like the water cycle?
Like the water cycle. Essentially, the formation and replication of RNA and DNA structures were thermodynamically favored because they increased the overall entropy production of the Earth system as a whole.
Wow.
The universe, in its relentless pursuit of maximal chaos, utilized life as its most sophisticated dissipator.
So we're talking about a kind of cosmic design here. The genetic code, the informational engine of all life, might be a successful secondary feature of a structure that was fundamentally optimized to be an energetic lightning rod. Physical persistence is just maximizing the flow of energy.
And that leads us pretty naturally to our second framework. If physical persistence is driven by maximizing energy flow on a cosmic scale, what does persistence look like on the microscale of, say, an individual human being?
We are pivoting now from Archean chemistry to the profound, spiritual, yet very systematic philosophy of Athathiri Maharishi and his concept of biomagnetism.
It's a radical parallel, but it's a parallel. The philosophy posits that universal magnetism is the foundational basic principle that underpins the entire evolutionary process of the universe.
And this universal magnetism, it isn't just a force like we think of it. It's described as an indivisible phenomenon made of two inseparable aspects.
Right, and this is where it gets a little abstract. So let's walk through the four core principles the source has defined.
Okay.
The first principle is the eternal space. This is the static, pervasive, unmoving backdrop, the void. The second is the energy particles, the dynamic, whirling, self-moving aspect within that static space, sometimes called the Holy Spirit in other traditions.
And the interaction between the static and the dynamic creates the third principle.
Exactly. Because of the friction and movement of these energy particles within the static space, they generate spreading waves. The sources call them shadow wave particles. These are the manifestations of the energy flow.
So the fourth principle, universal magnetism, is just all of that put together. The space plus the waves, the field of influence.
Yes, that's the cosmic engine.
So if that's the cosmic engine, then biomagnetism is our personal, localized instance of that engine.
Correct. Biomagnetism is defined as a pocket of that universal force, but one that's uniquely characterized by the laws of evolution to function specifically within the physical bodies of living beings. It's the dynamic energy that animates the system.
And this internal energy, it's not just sitting there. It's constantly transforming. The sources say it transforms into the 5-tan matrix. Pressure, sound, light or heat, taste, and smell.
And this conversion is the basis of our entire internal experience. In living beings, the physical transformation, the conversion, and then the exhaustion of this biomagnetism is precisely what results in all feelings. Pain, pleasure, peace, ecstasy. It's the currency of sensation.
Okay, here's the truly counterintuitive part. This challenges our everyday perception. We think enjoyment comes from the object itself. The source uses the example of eating a sweet cake.
We live under this illusion that the enjoyment, the sweetness, is in the cake. But the philosophy argues that the actual feeling of enjoyment is produced entirely internally.
By what?
By the induction, conversion, and consumption of one's own biomagnetism. The sweet cake is just the trigger, the inducer.
So the interaction with the cake raises the transformed degree of magnetism in my tongue, and that sudden flux of internal energy is what my brain registers as pleasure.
Yes. And when that magnetic energy is consumed, the feeling stops. The proof they offer is practical. If you eat something intensely sweet, which accelerates your magnetic transformation for sweetness to, let's say, a level 10,
Okay.
and then you immediately follow it with something mildly sweet, like coffee that's at a level 5, you can't taste the coffee's sweetness.
What?
Because your system is still operating at and subsiding from that 10-degree induction. It can't register the lower level stimulus.
That's a fascinating and testable claim. It means we're fundamentally just experiencing our own internal energy state, which is just mediated by external triggers.
And if we can control the internal energy, we can control the experience.
And this biomagnetism is also stored in what the philosophy calls the genetic center.
The genetic center is described as the central vortex of magnetic circulation. It's a complex psychic archive. It stores not just the physical blueprint, but the imprints of all deeds, experiences, and qualities acquired.
Both through heredity.
And by the individual during their own life. It's a living record of physical, mental, and energetic history, all encoded magnetically.
So how does this system explain thought and memory?
When we perceive an object, say a cow, the visual information is compressed into a microdot using biomagnetism. This magnetic impression is then taken to the genetic center and stored as a permanent quality or an imprint.
In recollection, thought.
That happens when the circulating biomagnetism strikes the relevant brain cells, which expands that microdot imprint back into a conscious image.
And this structure is what defines our destiny, through what the philosophy calls sin imprints.
Precisely. Every action, every thought generates these imprints, which then dictate future actions and results, manifesting as cycles of pain or pleasure. The purity of the genetic center dictates the future life cycle, which in this philosophy includes concepts like birth and rebirth.
So purifying these imprints isn't just a spiritual exercise, it's essential energetic maintenance for self-realization.
And for mastery over one's own persistent form.
Okay, now this is where I need to pose a challenge. We're talking about an energy storage mechanism, the genetic center, and energy transformation biomagnetism. In the physics section, the Tau model used very specific terms like ZNAT to describe a highly regulated energy budget for persistence.
Right.
How do the sources bridge this abstract concept of magnetic circulation to validated physical energy flows, like the chemical energy of ATP in our cells?
That is the ultimate connection we're trying to make here. While Vesasiri Maharishi's philosophy uses spiritual terminology, the function it describes, an energy system that collects, stores, and mediates internal fluctuations to maintain a complex structure, the body.
It's functionally identical to the CZ mechanism in Tau.
It really is. ATP is chemical potential energy. Biomagnetism in this model is the field that organizes how that energy is used. The core principle is the same. Persistence requires an internal buffer to survive external chaos.
And the practical tool for achieving this energetic persistence and purifying the genetic center is Kaya Kalpa Yoga.
Kaya Kalpa is an ancient rejuvenation practice, but it was specifically simplified by the Maharishi to be practical for modern life. It takes only 5 to 10 minutes a day. No medicines, no herbs.
So for a system that's dealing with, cellular deterioration and oxidative stress, how does a 5-minute routine actually achieve measurable anti-aging effects? What's the mechanism?
The primary mechanism is described as the transmutation of sexual energy. This focused practice directs and channels that vital fluid energy, which is seen as a highly concentrated form of biomagnetic energy, back up into the central system to revitalize the body's cells.
It's essentially recharging the battery.
Yeah.
Recharging that seasonal.
That's the idea. And secondly, it's said to enhance cellular polarity, which we know from biology is necessary for optimal ion exchange and overall cell function.
The benefits, according to the source, are they're really wide-ranging.
They're comprehensive. They include physical rejuvenation, better immunity, managing chronic diseases, mental benefits like improved memory and focus, energetic optimization of the whole biomagnetic field, and then the spiritual goal of self-realization.
This philosophy sees humans as the culmination of transformation, endowed with a sixth sense.
The ability to think and cognize the imperceptible, to move beyond just the simple sensory data of pressure, sound, light, taste, and smell.
And that cognitive ability is the mechanism for conscious energy control.
Exactly. The development of consciousness moves through defined stages in the yoga texts, from random thinking through various levels of focused concentration up to deep meditation. The ultimate goal is to master that internal energy flow, the biomagnetism, and purify the imprints in the genetic center.
And by doing that, The individual can align their own persistence with the source of all order in nature. It's persistence through self-mastery.
It is.
Okay, so we have a philosophical and energetic technology for internal persistence. Now, let's fast forward back to the laboratory and look at modern longevity research, where the focus is similar, controlling energy decay, but using hard cellular mechanics and rigorous intervention studies.
When modern science talks about aging, we're talking about cellular deterioration, chronic oxidative stress, and a steady decline in regenerative capacity. And one of the most powerful tools in this fight is the induced pluripotent stem cell, or iPSC.
Right. iPSCs are adult cells like skin cells that have been genetically reprogrammed back to an embryonic pluripotent state. But why are they so essential for aging research if they're basically baby cells?
The value is in the reset. The reprogramming process doesn't just make them immature, it effectively erases the epigenetic and molecular hallmarks of aging. It wipes the slate clean.
But that creates a paradox, right? An immature cell can't perfectly model an aged body. So what are the researchers actually studying?
They use that reset as a starting line. They study the molecular pathways that govern the reacquisition of age-related biomarkers. By starting with a youthful baseline, scientists can watch the process of decline begin all over again in a controlled environment.
So they aren't studying old cells. They're studying how youth is lost.
Precisely. And they can rigorously test interventions designed to stop or slow that decline.
Current anti-aging strategies are focusing a lot on cellular senescence, those zombie cells that stop dividing but refuse to die.
And instead, they just pollute the microenvironment with damaging signals. That's the senescence-associated secretory phenotype, or SASA.
Right, and therapies like SAR T cells are being engineered to selectively target and eliminate these damaged senescent cells.
It's a critical cleanup operation. It provides a healthier environment for the surviving cells.
But the sources stress a crucial limitation of just cleaning up the zombie cells.
Yes. While removing the bad actors is necessary, the surviving cells don't automatically rejuvenate. They don't get younger. They're just surviving in a cleaner house.
So true internal rejuvenation needs something more.
Yeah.
It needs metabolic mastery.
Which brings us to the crucial role of autophagy, that's cellular recycling, and the AMPK pathway.
Autophagy is the cell's own self-cleaning mechanism. It gets rid of damaged proteins and organelles. It's essential.
And we have some fascinating research showing a chemical way to induce this. A source discusses a compound called RRP. It was tested for its anti-aging properties, and it was shown to effectively attenuate cellular senescence that was induced by H2O2.
H2O2 being a common and powerful oxidative stress agent.
A very common one.
So how does RRP do this? What's the specific molecular lever it's pulling?
RRP achieves this anti-aging effect by inducing autophagy through the activation of the AMPK signaling pathway. AMPK, or AMP-activated protein kinase, is the central energy sensor of the cell. It's the master switch that monitors the energy budget.
That sounds conceptually very similar to the C-droid term in TELM, the thing that manages the internal energy buffer.
It's the biological analog, absolutely. When the cell's energy charge is low, when ATP stores are running, low AMPK activates, and that promotes processes like autophagy to generate more energy, while at the same time it reduces cell senescence.
And the research validated the inverse relationship with aging markers, right?
Completely. When they artificially aged the cells with H2O2, the levels of key factors in the AMPK pathway went down, and conversely, their downstream targets, specifically mTOR, became hyperactivated.
And mTOR, the mammalian target of rapamycin, its hyperactivity is closely linked to accelerating aging.
Right. And RRP reverses this whole cascade. It activates AMPK and shuts down mTOR, which demonstrates it's a really promising candidate for preventing things like endothelial cell senescence and atherosclerosis.
Okay, so we've seen pharmacological control over this metabolic persistence system. Now let's step back and look at the holistic mind-body interventions. This brings us back toward the kind of conscious control implied by Kaya Kalpa. Can we consciously manipulate this chemical engine?
The evidence suggests that, yes, we can. We have findings from the Yoga and Meditation-Based Lifestyle Intervention, YMLI study. It was a 12-week prospective study with 96 healthy participants, and it was designed specifically to test if a holistic intervention could impact cellular aging biomarkers.
What were the cardinal biomarkers of aging they were tracking?
They tracked oxidative stress and DNA damage. And the results were compelling. YMLI significantly reduced the DNA damage marker 8-OH2DG, and it also significantly reduced reactive oxygen species, or ROS.
And at the same time, it boosted the body's defenses.
Yes, significantly increasing total antioxidant capacity, or TAC.
But the big headline grabber result always involves telomeres. Did YMLI actually reverse the biological clock in that sense?
It impacted the mechanism of the clock. YMLI significantly increased telomerase activity. Telomerase is the enzyme responsible for maintaining and lengthening telomeres, those little protective caps on our chromosomes that shorten every time a cell divides.
So increasing its activity is a direct countermeasure against that decline.
It is. However, and I know you recall this, the sources noted a really important statistical nuance regarding telomere length itself.
Right, this is a critical detail for a deep dive audience. The activity of the enzyme that lengthens telomeres went up significantly. But the actual measurement of the average telomere length, while it did increase, the finding was not statistically significant. The p-value was 0.069.
Yeah.
So let's pause on that. Why is the statistical non-significance of length, even though the repair enzyme activity went up, why is that a common challenge in telomere research? What does it tell us about the intervention?
It tells us a couple of things. First, telomere length measurement is just inherently noisy, and it takes time to change dramatically. But second, and I think more importantly, it suggests that the cell is prioritizing maintaining its current length and repairing damage.
It's a stabilization effort.
Exactly. The cell is stabilizing its structure. It's investing energy into the repair crew telomerase, even if the overall length hasn't moved the needle enough to be statistically certain in just 12 weeks. The biological reversal is happening at the level of function and defense, which is arguably more important than just the simple length.
And beyond structural repair, the YMLI study also hit the metabotrophic biomarkers, the hormonal and inflammatory markers.
It did. The intervention significantly reduced stress hormones like cortisol and the primary inflammation marker IL-6. That's a massive reduction in chronic stress load.
And on the positive side,
It significantly increased factors like beta endorphins, BDNF brain-derived neurotrophic factor, which is essential for neurogenesis, and sirtuin-1, a protein strongly associated with metabolic health and longevity.
this comprehensive, multidimensional control over the internal state is what separates this holistic approach from just single interventions, like taking a pill.
The sources really drive this point home. A single intervention, like taking antioxidant supplements, might only decrease ROS. And that can paradoxically shorten your lifespan because your body actually needs a certain level of ROS for things like immune response signaling.
But YMLI regulates ROS.
It regulates it through the balanced management of stress-related processes and gene expression. The integration of the physical exercise, the breathing practices like in Kayakalpa, the meditation, the introspection, that's the key to managing that delicate energy budget. This is wars of the human system.
Okay. We've established that persistence, both at the origin of life and in longevity, depends on actively capturing, storing, and channeling energy to maintain structure. Now let's go to the highest level of structure, the entire body plan. We're moving from controlling cellular function to controlling anatomical form through this non-genetic master control system, bioelectricity.
This is where biology starts to look like engineering software. Bioelectricity research suggests that the electric fields generated by cells are the true master regulator. They're strong enough to actually override chemical signals.
How fast are we talking?
We're talking instantaneous. When a cell is damaged, its membrane potential changes immediately. Ions pour out, creating a wound-induced electric current that flows instantly across the tissue way faster than any chemical signal could ever diffuse or be synthesized.
So if I cut my hand, the electric field is the general in charge, instantly directing the entire healing process. And the chemical messengers, the cytokines, the growth factors, they're just the logistics team following the electric orders.
That's a great analogy. It holds up well. Researchers have tested this, and the electric field consistently overrides the other factors. It acts as the primary blueprint coordinator. And this led to a profound hypothesis. Pattern memories, the instructions for what shape the body should be, are encoded in electrical circuits, not just genes.
And this is where we have to talk about the famous two-headed worm experiment with the planarian flatworms.
Planarians are exceptional. They're essentially immortal, they're asexual, and they have this incredible regenerative capacity. And scientists have noted their genomes are often a mess, which has led to the suspicion that DNA, the sequence itself, might be a bit misplaced as the sole blueprint for their persistence.
So tell us the mechanics. How did they convince a worm fragment to grow two heads instead of a head and a tail?
The researchers took these planarian fragments and they briefly dipped them in a simple chemical solution. Now, this chemical didn't modify the DNA at all. It just temporarily blocked the function of gap junctions.
Those are the channels that let the electrical signals pass between cells.
Right. By blocking that communication, they briefly disrupted the body-wide electrical circuit that defines the normal, one-headed plan.
And the result of that fleeting exposure was a permanent structural change.
Precisely. The transient disruption caused the fragments to regenerate into two-headed animals. But here's the part that just blows the doors off traditional biology. This structural change was permanent and heritable.
Wait a second. So they took that two-headed worm, they cut it up again, and because the original chemical exposure was fleeting, the new fragment should have reverted back to the default one-headed state. Right.
They should have, especially since their gene expression was normal and the chemical was long gone. But when these genetically normal worms were recut, they continued to produce two-headed offspring. That temporary electrical change made a permanent, non-genetic memory that was inherited.
The structural blueprint became an inherent trait, passed down, even though the chemical that caused it was gone and the genes were never changed.
The conclusion is that the anatomical pattern, the persistence of the two-headed form, it's not stored in the individual cell's DNA. It's stored in the body-wide electrical circuits. It's been described as a cognitive glue, or a collective intelligence of cells navigating anatomical space based on voltage gradients. The anatomical persistence is fundamentally software encoded electrically.
This just deeply challenges the gene-centric view of evolution, the modern synthesis, where acquired characteristics can't be inherited. This takes us right into the realm of epigenetic inheritance.
Right. Epigenetic inheritance refers to traits acquired by parents that influence their offspring without altering the actual DNA sequence. Historically, this aligns with ideas similar to Lamarckism.
So if the DNA sequence isn't changing, what is the non-genetic blueprint being transmitted by?
The molecular mechanisms are largely mediated by non-coding RNAs, or ncRNAs. These are molecules that don't produce proteins, but they act as powerful regulators. You have short ones like miRNAs and piRNAs, long ones called LNcRNAs. They're the switches and the rheostats of the genome.
And these switches are highly sensitive to the environment and the parental state.
Absolutely. The sources confirm the massive transgenerational impact of environmental factors. Parental stress, diet, alcohol exposure. They can cause epigenetic programming in the offspring that affects complex traits like stress response, anxiety, metabolic disorders.
So the stress of the father, for example, can predispose the child to anxiety through molecular changes in his sperm, totally independent of the mother's experiences.
Exactly. Just like the bioelectric circuits hold a pattern memory, the ncRNAs in the gametes hold an ancestral environmental memory that dictates future persistence and vulnerability.
That's why the field has developed this therapeutic approach called enviromimetics.
It's a preventative approach. Since the environment is the epigenetic regulator, enviromimetics suggests consciously ameliorating paternal conditions, addressing stress, diet, lifestyle before conception to prevent or even revert these epigenetic alterations in the sperm.
It's conscious, proactive control over the non-genetic blueprint of the next generation's persistence.
It is.
So the ultimate goal then, if we can control this bioelectric software and this epigenetic blueprint, it's to completely define our own physical persistence. We can choose our own form.
This is the logical extreme, and researchers are already building synthetic life forms. We have xenobots made from epithelial cells that can self-replicate kinematically. We have anthrobots made from adult human tracheal cells that can navigate and heal neural wounds in a dish.
And these are described as vehicles for studying the contents of latent space.
Latent space, meaning the realm of all possible biological forms the electric code will allow.
Wow.
And the theoretical vision is the development of an anatomical compiler, a sophisticated bioengineering tool where a user could specify the precise living form they desire, a body with better immune function, greater regeneration, specific cognitive enhancements.
And the compiler would just output the exact sequence of bioelectric stimuli needed to tell the cells to build that specific structure.
That's the vision.
This represents the ultimate aspiration of persistence, freedom of embodiment.
It does. The advocates of this research suggest that future generations will look back at us and pity us for being stuck in the random walk of evolution, forced to suffer the degeneration and the random genetic flaws and the inevitable decline of aging.
In that future, humans could choose their IQ, their physical capabilities, their lifespan, overriding the chaotic elements of our existence.
This is the complete victory over entropy. The self-aware agent choosing its own path toward physical perfection and persistence, effectively telling the universe, no, I will not succumb to chaos yet.
That thread of energy management connects everything we've talked about. We started with the physical necessity from Tal, that life must emerge just to dissipate energy.
Then we explore the ancient mastery from Kaya Kalpa of capturing and storing that internal energy.
And we concluded with the bioelectric mastery of using that energy to reprogram the persistent anatomical form itself.
Life, in all its forms, is fundamentally defined by its ability to capture, store, and channel energy flow effectively to maintain an organized structure against decay.
So the great conflict this deep dive reveals is the ultimate question of life's goal.
Is life's goal defined by an ancient, purely physical necessity to simply maximize entropy dissipation, like the cold physics of Talam suggests?
Or is life a conscious, self-aware navigational tool that can be steered and perfected through bioelectric and energetic mastery, like Kayakalpa and the anatomical compiler vision suggest?
If the human body, this collective of trillions of cells, operates under a kind of cybernetic goal to build and maintain a persistent body plan, and if the tools of physics and bioelectricity give us the power to perpetually reprogram and perfect that goal, what happens to human consciousness when it finally achieves its ultimate anatomical purpose? Perfect physical persistence.
That is the provocative thought for you to consider today. If we achieve ultimate freedom of embodiment, if we eliminate all biological struggle, all degenerative pressure, all need to fight against the random walk of evolution, what does consciousness even strive for when physical persistence is guaranteed for a billion years?
Would the absence of a thermodynamic or a biological challenge lead to stagnation? Or would it finally free us to pursue a truly new, entirely different goal?
Think on that until our next deep dive.